Hey, thanks for the introduction and giving me the opportunity to present my work. So all cells in our bodies need to take up nutrients and remove cell signaling receptors from the surface in order to regulate signaling. And one major pathway to allow cells to do this is clathrin-mediated endocytosis. And I'm especially interested in how the actin cytoskeleton functions in this process. Actin filaments are linear polarized polymers that preferentially grow on one end, which we call the plus end. And when they grow, they can produce a force against an object, for example, the plasma membrane. And this force can be enhanced by the ARP2/3 complex, which can bind to the site of existing filaments and induce the formation of a new filament. We then call the old existing filament the mother filament, and the new filament a daughter filament. And these branched actin networks play many different roles inside mammalian cells, for example, in cell migration or in membrane trafficking. And we are particularly interested in how these function in clathrin-mediated endocytosis. We use clathrin-mediated endocytosis to understand mechanisms of actin force production in inside human or mammalian cells. During endocytosis, the relatively flat plasma membrane is deformed to generate a vesicle that contains the cargo. And in mammalian cells, the membrane is under tension, which counteracts this deformation process. And deformation is enabled by the formation of a clathrin-containing coat. And after initial transition into this U-shaped plasma membrane form, the membrane is further deformed in such an omega shape, which then allows a small GTPase to bind to the neck of these endocytic sites that then facilitates scission of the vesicle. Importantly, these clathrin codes can be identified by electron microscopy due to their characteristic hexagonal and pentagonal shapes. Besides of the clathrin code, we also know that we need the branched actin machinery to support these membrane deformation processes. But we don't understand how these actin networks actually organize in order to produce force during this process. Previous work showed that the activators of the ARP2/3 complex actually localize in a ring around the base of these endocytic sites, and that there are actin binding proteins inside the coat, such as this rod-shaped protein HIP1R. My colleague in the lab, Matt Akamatsu, used this information to set up a mathematical model, and we can see the first frame of a simulation of one of his simulations here. So we have an endocytic site in the center. In purple, we have these HIP1R molecules at the ring around this endocytic site. We have pre-activated ARP2/3, and we have free-floating short actin filaments inside the cytoplasm. When I now start the simulation, what we found is that this minimal these minimal components are sufficient to allow actin to self-assemble into a branched radial array that binds to these endocytic sites and grows against the plasma membrane on top and produces a net pulling force that pulls down these endocytic sites into the cytoplasm. We had to assume several of the parameters that went into the model, and there's several testable predictions that now arise from the model. We want to know whether there's really this branched array that self-organized that produces force by polymerization to induce membrane deformation. We wanted to understand how this branched actin formation is initiated. So what's the source of these mother filaments and how are they organized? And then I will talk about what the precise localization of these actin linkers, this HIP1R molecule is, and what's the impact of the localization on CME-actin organization and function. An ideal method to address these questions is cryo-electron tomography, which is a 3D electron microscopy method. And for electron tomography, we directly grow our mammalian cells on EM grids. And one important thing we have to make sure is that these cells spread thinly across these grids, because we can only image very thin regions of these cells due to the poor penetration depth of the electrons in the microscope. After the cells spread on these grids, we rapidly freeze our samples by blunt freezing. It's important that we only blot our samples from the back in order to not to destroy or remove the cells from the grids. Then we can directly use these samples for tomography imaging. One disadvantage is doing it this way is we would basically shoot blind into the cytoplasm. We can't really see anything. We can't be sure that there's an endocytic site in there before we reconstruct our tomograms. To get around of this problem, we can use cryofluorescence microscopy, where we use fluorescent markers to target a specific region of interest. But most of the tomograms I will show you were recorded randomly inside the cells. So we then can identify different protein codes inside the cells. And here's an example of a coated vesicle. So we have the vesicular area in this yellow circle here, and then the protein code around. If we now zoom or go down or up in Z position, we can see this characteristic cage-like structure, which looks like the clathrin-coated that was observed by other electron microscopy methods before. But to really prove protein identity, we need to have higher resolution, and this can be achieved by subtomogram averaging. And what we did in principle is we extracted these vertices of these cages and then averaged across multiple tomograms of these. This allowed us to get the first native clathrin-coated structure, and this structure was sufficient, or its density is the EM map is sufficient to place a previously published cryo-EM structure into this density, and the subtomogram average has a resolution of 2.7 nanometers. Just to add here and give you an idea where this method is moving, there were 3 reports that using subtomogram averaging inside cells, sub-nanometer resolution is actually possible, and we will hear more about one of these stories in a later talk today. So the nice thing about the system here about using this in situ cryo-electron tomography is we can now put back this information into the original 3D space inside the cytoplasm of a cell. So this is a movie of a tomogram, but you will see when I start the movie, there will be this clathrin-coated vesicle from the slide before showing up in the center. So there's the vesicle. And what we can see in the center of this vesicle is that there's a tail of many different actin filaments. Now we can place back these subtomogram averages into the 3D space and manually segment these actin filaments, which allows us to analyze the spatial organization and relation of these actin filaments and the different clathrin codes inside the cells. We obtain tomograms of different stages of endocytosis. We can so we can classify them based on the membrane shape. So flat CME sites or early CME sites, these invaginated sites, and these clathrin-coated vesicles I just showed you before. And here are the corresponding segmentation models. I produced them together with my undergraduate student, Amir, and we just helped to reduce bias during the manual tracing. Surprisingly, what we found is that there was always a mix between branched and unbranched filaments. So these networks were not exclusively made of branched actin filaments. And there was an increase in branching towards later stages. And what we think what's happening here is that these unbranched filaments are just pre-existing actin filaments. And then after activation of the up-to-3 complex, there's a gradual increase in branching happening during endocytosis. Then next, we wanted to test something that's called the single mother filament theory, which is depicted in this cartoon here. And it's based on platinum replica EM of unroofed cells. And the theory or the idea behind is that all branched actin during CME actually originates from a single mother filament, which is in contrast of what we found, which is that in all our tomograms, we see that the branched actin is actually organized in clusters, which is consistent with our mathematical model. And our conclusion here is that branched actin actually originates from multiple founding mother filaments that initiate this branched actin formation. Then we wanted to analyze the force-producing capability of these actin networks that we observed. And we set up two different methods that allowed us to analyze the polarity of these filaments. And we also came up with new software tools to display the polarity analysis. So what we found is that these actin filaments either grow up to the plasma membrane or towards these endocytic sites, or more precisely towards the neck, which is indicated by the red color here. And this filament orientation towards the neck came a bit to a surprise to us because that's not something we found in our model before. And we were wondering what could be the reason for this growth against the neck. So from the model, we knew that one of the major geometry driving components is this molecule HIP1R. And we were wondering whether HIP1R could play a role here. So HIP1R forms these rod-shaped dimers, which have two globular actin binding domains on one side and two globular membrane binding domains at the other side. Then we searched our tomograms to see whether we can identify densities that look similar to this. So we have 3 tomographic slices here that correspond to these boxes on the segmentation model here. And indeed, we could find something that molecules or densities that really look like these HIP1R molecules here that were from these old EM pictures. To further test whether this indeed could be HIP1R, we applied subtomogram averaging to this putative actin binding domain, the cytoplasmic actin binding domain. By doing this, we could get a low resolution density map, but this low resolution density map was sufficient to place 2 actin binding domains and the dimerization domain of a homologous protein, which is called talin. So this further supports the idea that this could indeed be HIP1R. Surprisingly, what we found is that HIP1R, or this molecule, doesn't only localize to this bud of this endocytic site, as we assumed before, but we can also find these densities around the neck. And we were wondering what could be the density of this neck localization. As I told you before, the main geometry defining boundary conditions for actin and clathrin-mediated endocytosis are the ring organization of the activated up to 3 at the plasma membrane and the position of these HIP1R molecules. If we now place HIP1R also at the neck of these endocytic sites, what we can see is that these actin filaments now wrap around the neck, where they potentially also produce a squeezing force that, besides of aiding in internalization of this endocytic site, could now also help in squeezing the endocytic neck. Moreover, this neck localization also enhances or improves internalization and efficiency by about 50%. To sum up what I told you today, we found that endocytic actin self-organizes into a branched arrays that produce force by polymerization. We found that branched actin is initiated by multiple founding mother filaments. And we found that these branched actin filaments are oriented towards the base of the pit, these endocytic sites, but also towards the neck of these endocytic sites. And we further found that the position of the HIP1R linker at the neck increases endocytic internalization efficiency, and also Yeah, thanks. Yeah, so increases internalization efficiency. With that, I want to thank members of the Trubin Barnes Lab and Davis Lab at UC Berkeley. A big shout out to my colleague, Akamatsu. I've been collaborating with him for the last 3.5 years or so. This collaboration really helped both of us because we could really accelerate the progress of both our projects, which wouldn't have been possible if we would have taken an individual approach. So by joining our forces, we were really able to push forward both our projects. And then I want to thank our collaborators in the Rangamani lab at UC SD and all people in our electron microscopy facilities. And if you want to reach out to me and have further questions, you can email or reach me on Twitter. Thanks for your attention, and I'm happy to take questions.